In this video, we're going to talk about the movement of water and small solutes across the capillary wall. Now, if you recall, we did discuss the movement of water across cell membranes previously, and that was driven entirely by one force, osmotic pressure. And so at the lower right here, I have a cell where I've drawn some solutes on the inside, and in this scenario, a higher concentration of solutes on the outside here. And because of that, we have an osmotic gradient, a concentration gradient, which is an osmotic gradient across that cell membrane that would tend to drive water movement out of the cell, concentrating its solutes to the point at which they would actually match that of the interstitial fluid. And so there's really no standing osmotic gradient across cell membranes, but it is constantly tweaked by the fact that we eat solutes and we drink water and we upset that balance all the time. We just tend to reestablish it rapidly through the movement of water across aquaporins. Okay, well, when we start to look at the capillary barrier, we still have an osmotic pressure gradient, but now we have a secondary driving force. And that secondary driving force is this blood hydrostatic pressure. And blood hydrostatic pressure is generated by the heart. We compress the blood and push it into the blood vessels, and that pressure diminishes with distance along the vascular tree, along the blood vessels, due to friction. Now, so that is indicated by the size of the arrow, these outward arrows sort of falling as we go from left to right and flow along this top capillary, this perfused capillary. Now, that's going to tend to, that hydrostatic pressure not only tends to drive blood flow down the pipe, but it also tends to generate what's called a transmural pressure. It also pushes outward on the walls of that blood vessel, tending to drive what we call filtration, or movement of water and small solutes across the capillary barrier. Now, when we talked about cells, it was water only because the cells were relatively impermeable to small solutes. Capillary walls are much more permeable. We have these gaps between the cells, and the glycocalyx that lines those cells is actually very permeable to the small solutes. And that's why it's called bulk flux, because water and solutes move in bulk across that capillary barrier. Now, we do have an opposing force, and that is this blood oncotic pressure, which is the blood osmotic pressure due to large solutes. And the reason we are specific there is that the large solutes are the only trapped solutes. In other words, blood has a very high osmotic pressure, but it has a very low tonicity, meaning most of the solutes are permeable, only these large protein solutes are trapped. So that's really the only osmotic force we really need to recognize, because that's the one that can maintain a gradient, because those solutes, blood solutes are trapped, the protein solutes are trapped and are reflected, they bounce off those walls and they stay on the inside. And so we have a standing oncotic pressure that drives water and small solutes inward. And so the net movement across the capillary wall is due to the balance between, or lack thereof, between an outward hydrostatic pressure and that inward oncotic pressure. And as you can see at the top here, we have a perfused capillary, meaning blood is flowing through it. And in that scenario, the blood hydrostatic pressure tends to win along the entire length of the capillary, driving the filtration that we see here by these arrows at the bottom, right? Driving this fluid and small solutes out into the interstitial space. And a portion of that would be taken up by the lymph vessels. Now, that sounds like it would be a lot of filtration. However, we also have many capillaries that are unperfused at a given moment, right? Capillaries tend to transition back and forth between this perfused and unperfused scenario due to constriction and relaxation of the smooth muscle that precedes these capillaries in what are called the arterioles. And that helps maintain homeostasis of those tissues, but one of the side effects of that is that these unperfused capillaries have a much lower blood hydrostatic pressure, right? They don't have blood flowing through them, so that outward pressure falls dramatically. And as you can see down here at the bottom, our inward arrows now win, right? The blood oncotic pressure arrows win, and we get net movement of that filtered fluid from the top capillary moving into an adjacent capillary, or capillaries, that are unperfused. And the two tend to nearly cancel each other out. In other words, the filtration and the reabsorption by other capillaries tend to almost cancel each other out. There's a little bit of extra formation of interstitial fluid that becomes lymph, and that is actually returned to the blood up in the thoracic cavity. And that's important to the circulation of the interstitial fluid, that we generate this little bit of extra interstitial fluid. In the lymph system here, it gets exposed to white blood cells, which is very useful for immune protection and so on.